Yeah, I'm Hongryul Ahn, an assistant professor, Division of Data Science, the University of Suwon from Korea. I participated in the CAFA challenge as one-man team. And now I'm talking about a research related to disorder gene prediction. That is one of the CAFA4 challenges. And the title of my research is Network Propagation Embedded Deep Neural Network for Disorder Gene Prediction. Disorder gene prediction is a research topic, and network propagation embedded means a feature selection method, and deep neural network indicates an inference model of disorder prediction. Let me get started. First, I introduce a background of this research, DO, that is disorder ontology. Disorder ontology is a set of biological knowledge about which gene is involved in which disorders. Disorders in DO are arranged into 130 DO IDs with hierarchical structure. The yellow box shows an example of 4 disorders in DO. And the DO annotation known so far are publicly available from this link. And DO association of a gene can be represented as a multi-up vector. For example, let there be a gene GA, and the DO annotation of GA is represented like this. The value 1 in the first column means GA is known to be associated with DO1, and the value 0 in the second column means GA is known to be not associated to DO2, or GA is yet unknown whether it is associated to DO2 or not. The DO annotation is useful information because it tells human being which gene is involved in which disorders. However, there is a problem. Problem is that most of association between gene and DO are not unknown. The figure shows an example of DO annotation vector between 130 DOs and N genes. And in the figure, 0 values in the blue boxes shows a lot of unknown association between DO and gene. So the reason why is incompleteness of our knowledge. Thus, to illustrate the unknown DO association, we have to predict them from the other gene feature information. And this problem is called DO inference. DO inference helps us to discover new knowledge for the function of genes. Then the DO prediction is selected one of the CAFA challenges. And this slide shows the detail of DO inference problem. DO inference is regarded as a kind of machine learning problem that infers the DO of a gene from gene feature data. For example, the input of DO inference problem is a gene with a gene ID and its amino acid sequence. Then we extract features of the gene to generate the data of a gene by investigating sequence feature, functional feature, or relationship information to other genes. Then from the data of a gene, we can infer DO and produce a DO predictive label. This is the DO inference problem. And this slide shows research challenges and contributions. There are 2 challenges of DO inference research: feature extraction and inference model. Feature extraction is a process to select features of genes to be used for the inference. For feature selection, I collected gene information from gene database, and especially I utilized edge information to improve DO inference. To utilize edge information, I used network propagation algorithm. Also, for inference model, I used deep neural network. That is the most popular machine learning model recently with binary cross entropy. Now, I will show you the method of my research in detail. And this slide shows the overview of my method. The method consists of 2 major parts. The first part is feature selection part, the left side of gene feature vector. And the second part is DO inference part, right side of gene feature vector. The goal of feature selection is to build gene feature vectors from gene databases. To build the gene feature vectors, I developed two sub-methods. Sub-method 1, gene feature selection, collects gene features from gene databases and fill the values into gene feature vectors. But the gene feature selection do not use gene relationship information. Thus, sub-method 2, network propagation feature selection, utilize gene relationship network to generate propagated gene features. Finally, gene feature vectors have large dimensional features consisting of gene feature and propagated gene features. After building gene feature vector data, sub-method 3, DO Inference, infers DO labels from gene feature vectors using deep neural networks. I will explain you the three sub-methods one by one. And the first sub-method is gene feature selection. The goal of gene feature selection is to extract gene features from gene database. To do this, gene feature selection collects gene information from gene database and produce gene feature vectors. This is an example of gene feature vector that is the result of gene feature selection. See the example gene feature vector of G1. This vector consists of about 50,000 dimension features in total. The features are originated from 4 types of database: CDD, GO, and amino acid proportion, sequence length bins proportion features. CDD features occupies 34,000 dimension, GO about 16,000 dimension, amino acid proportion 24 dimension, sequence length bin 13 dimensions. I will explain them in detail. First type is CDD. CDD means conserved domain database. Conserved domain means a highly informative sequence pattern that are conserved across multiple species. There are about 34,000 conserved sequence patterns. Then I downloaded them from the NCBI site and conducted breadth search of the CDD patterns to the gene sequences. Then I assigned Boolean values. 1, if a gene has a CDD pattern or on its amino acid sequence, or 0 otherwise. In this way, I fill CDD Boolean values in about 34,000 CDD feature dimension. Second type is GEO. GEO, the full name is gene ontology, is a category of gene function. And there are more than 16,000 GEO identifiers. Then I downloaded them from the Unipro site and I assigned Boolean value 1 if a gene has the GEO identifier or 0 otherwise. In this way, I fill the Boolean value in about 16,000 GEO feature dimensions. Third, amino acid proportions. Amino acid proportion is the proportion of amino acid within a gene sequence. After investigating the proportion of amino acid, I assigned the float values into 24 amino acid feature dimensions. Fourth, sequence length bins. Sequence length bins means the length of amino acid of sequence. After investigating the length of gene sequence, I assigned Boolean values according to the sequence bin. 1 if the length of gene corresponds to the bin, or 0 otherwise. There are 13 sequence length bins whose intervals are shown like in the gray box. However, there is a problem. The problem is the most of values are 0. The values in the dimension are very sparse, especially in the case of CDD or GEO. So this is the problem I want to solve. So I overcome To overcome zero value problem, I developed the second sub-method, network propagation feature selection. In this sub-method, the network propagation algorithm convert the black and white values, gene feature values, into smoothed range values. In the network propagation, gene network is utilized. The propagated gene values then are appended into the gene feature vectors. Network propagation is an algorithm that smoothing feature values. It takes a graph G and a vector of node value P0 and a constant restart rate E. Then it produces a vector of node values PT by propagating the original input node values along the graph as step T. The detailed mathematical formulation is like this. See the example. Only one, in this figure, only one gene has non-zero value and the other gene have zero values as T0. Then the value of D is propagated to the neighbor nodes at the next step and the next step. In this way, the one-half value are all propagated to the entire neighbor nodes according to the network propagation algorithm. So network propagation is a way to smoothing black and white values into gray ranged values. For performing network propagation, we need a gene network. This slide shows a method to construct a gene network. To construct gene network, I developed network construction method called sequence similarity gene network construction. First, sequence similarity gene network construction runs BLAST for all pairs of sequence of genes. Second, it initializes a graph G as an unconnected graph. Then, for GI in the gene set, it adds connections between GI and each n nearest neighbor genes into a graph G, where n nearest neighbor genes are in the same species of GI. And continuously, it adds connections between GI and each m nearest neighbor genes into graph G, where m nearest neighbors are in the other species of GI. In this way, m plus m nearest neighbor graph, nearest neighbor gene network is generated, finally. And by using network propagation, an extended gene vector is produced. The original gene vector has about 50,000 dimensions, but they contains a lot of zeros values, especially for CDD or geo features. So I generate the smoothed value by performing network propagation with a restart rate ratio 0.1. I perform network propagation on 4 different gene networks. The 4 different gene networks are generated by different N or M options as shown in the figures. Yellow, green, pink, gray, like this. And the result of network propagation are concatenated to the gene feature vectors and resulting in extended gene vectors. So as the result of sub-method 2, the dimension of extended gene vectors increased 5 times than the original gene vectors. And most of values are added are non-zero. And as the result of sub-method 1 and 2, we now obtain gene feature vectors. The sub-method 3 in the red color is to infer DO to produce DO label vectors from gene feature vectors. For the inference model, sub-method 3 use deep neural network. This slide explains machine learning procedure for DO inference. First, it splits genes into training and validation dataset by 4:1 ratio. So 78 genes are used for training dataset and about 15 genes for validation dataset. And second, it builds a deep learning model. The structure of a deep learning model will be shown in the next slide. Third, it initializes the best model B as null value. And fourth, for 400 epoch, it trains the deep neural network model using training dataset with the batch size of 256 and loss function as binary cross entropy with L2 normalization. And then it calculates loss function value using validation dataset. And it updates the best model B if loss function is minimized. Finally, it infers DO for all genes using the best model B. And this is the structure of a deep learning model. I used the multi-layer perceptron, where input dimensions are about 252,000 and the output dimensions are 130. The deep learning model consists of 4 hidden layers, including 600 hidden nodes in each layer. And it uses ReLU activation function and L2 regularizer in the hidden layer. And for the last layer, the sigmoid layer is used. And this is the implementation and experiment environment. The experiment code is implemented by Python 3 Keras library. And the experiment is conducted on non-GPU machine because of large size input data. The data size is 45 gigabyte as of 32-bit encoding. So it cannot be run on GPU machine. So the experiment is conducted on workstation with a large memory machine. The spec is 64 cores, 128 threshold thread CPU, and 512 gigabyte RAM. And now I will show you the result of my research. The CAFA4 challenge of DO inference is progressed along the timeline. At the first phase, the target released in detail 97 genes and IDN sequences from 17 species were released. The second page, each of competition team submit their DO prediction result for the target gene to the online system. At the third page, CAFA assessor carried out evaluation to present it in ISMB 2012, and I was selected as presenter. And this slide shows input data and submission of DO inference challenge. The input data is 97 target genes with gene sequence, and the submission is DO prediction result for the target genes. The figure shows an example of submission. The first, second, and third columns represent gene ID and DO ID and predicted scores. And each submissions are evaluated by Fmax metric. Fmax is a modified version of F1 score. F1 score measures correctness between predicted set and true label set. But because of the submission is rank-based result, so F1 is not able to be applied directly. So Fmax use a lot of threshold T to determine predicted set result. Red box shows the predicted result determined by threshold T equals 0.36, and blue box shows the predicted result determined by the threshold t equals 0.11, and testing a variety of threshold value t, the best F1 score is selected as Fmax score. And this is the evaluation result of DO Inference challenge. My method showed the highest Fmax value in both of full mode and partial mode evaluation. And I skipped the explanation of full mode and partial mode. And this is the summary of my presentation. My research of DO Inference makes two contributions. I developed feature selection method that collecting gene information from gene database and especially utilizing gene edge information using network propagation techniques. And I developed inference model based on deep neural network with binary crest entropy. And this is my end of my presentation. Thank you for